In this video, we will discuss about this NY-ESO expression changes with DiaBZl treatment. In the previous video, we have seen that in the Western blot results, there is no change in the NY-ESO-1 expressions on the tumor cells when they are treated with the DiaBZl even after 3 hours and 6 hours. However, when they use the different detection technique, that is using the immunofluorescence, they can see the changes in the NY-ESO expressions on the tumor cells with the DiaBZl treatment at different time intervals. So the panel A is the results that is shown, the quantifications from the Western blot that we have seen in the previous video, or I have just shown you, that is these Western blots. And here we see that DiaBZl's expressions is not statistically significant. The NY-ESO expressions is not statistically significant between the control and the DiaBZl treatment after 3 hours and 6 hours. However, when they check using the immunofluorescence, they can clearly see the difference. That is, when the tumor cells are not treated with the DiaBZls, they do not see the NY-ESO expressions, but after 3 hours they see that NY-ESO expressions, and it further enhances in the after 6-hour treatment of these DiaBZls with the tumor cells. And these things are quantified in the panel C. We can clearly see that after 3 hours the expressions of NY-ESO-1 is high, whereas after 6 hours it goes down, indicating that the antibodies that are used for immunofluorescence will be able to detect this NY-ESO-1 more with more efficiency or with more accurately compared to the Western blot that they have done for this NY-ESO-1 expressions. And apart from this immunofluorescence, they also check this NY-ESO-1 and other antigens that are expressed on the tumor cells, that is HLA-A, using the flow cytometry. And what they have find out is that after 3 hours they can see, what they have find out is the same results that they have observed in the immunofluorescence. That is, the panel D shows about this NY-ESO expressions after 3 hours, where they can see there is a shift in the DiaBZls compared to the control, and indicating that there is an increase in the DiaBZls. There is an increase in the NY-ESO-1 expressions on the tumor cells compared to the control, where the tumor cells are not treated with DiaBZls. And for the 6 hours also we see that increase. That is, for this DiaBZl treated tumor cells has more NY-ESO-1 expressions compared to the control. And the other antigens has also shown the changes. That is, for the 3 hours the HLA-A02 shows a significant increase, but for the 6 hours this increase is there, but it goes down. And these things are also in line with the quantification results for the NY-ESO-1 expressions, where we can see that for the 3 hours the expressions of the NY-ESO is high, but it goes down after 6 hours. And this is also reflected in this HLA results. So, however, they have not shown the quantification results of this flow cytometry in the main figure. But when they check this NY-ESO expressions in the tumor cells that are not treated with the DiaBZls, and in the control case they do not see any changes in the increase in these NY-ESO expressions. And these results are shown in the supplementary figures. You can go through those supplementary figures from the reference that has been provided in the description. So from the immunofluorescence and the flow cytometry, they made a point that NY-ESO expression increases with DiaBZl treatments of the tumor cells, has been indicated by the immunofluorescence and the flow cytometry. However, this Western blot is not significant or is not sensitive enough to differentiate those increase in the NY-ESO expressions in different conditions. So I hope you understood how they made their point about NY-ESO expressions changes with DiaBZl treatment. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.